Hi, before I start off, I would like to present to you an example on identical twins that is related to epigenetics.
Imagine you have identical twins, and it is proven that the identical twins have exactly the same sequence of DNA. They were separated soon after being born. One remained in Singapore, while the other is sent to a poor and rural farming area. Imagine meeting each other after 30 years. Will they actually look the same? High chance, not really. The twin who reside in Singapore is likely to be more affluent and well-nourished. Therefore, he might be taller than the farmer. However, the poor farmer might be more toned-looking due to the labourer's work, while the man in Singapore might be fatter due to the stressed lifestyle. However, if we extract their genes, it is still the same as when they were a baby. But why the differences? One of the answers is epigenetics, which literally means above genetics. Epigenetics is not about changing the DNA sequences, but it is about how much or whether the genes are expressed in different cells of the body. It is just like how the brain cell of you and me actually differs from our heart muscle cell, even though they contain the exact same DNA blueprint. But before we zoom into the mechanisms behind epigenetics, let's introduce some background information of DNA organization to help you better understand epigenetics later on. Take a look at the location of DNA. DNA are organized into chromosomes, which are located in the nucleus. Chromosome is the structure within cells that contain genetic information. It comprises of DNA and protein, for example, histones. So how does DNA compact into chromosome? Starting off with the DNA is wrapped around histones, and further compaction occurs by binding to other proteins to form a chromosome. So how does DNA become useful information? We can explain it through the central dogma of molecular biology, which is the flow of genetic information, from DNA to RNA to make a functional product, a protein. To make protein from DNA, it first needs to take an intermediate step. That is to make ribonucleic acid, RNA in short, from DNA by transcription. So DNA acts as a blueprint read by RNA polymerase, which produces a RNA strand, followed by translation. In this process, mRNA is decoded and translated to produce a polypeptide sequence, otherwise known as a protein. Proteins are main contributors to different shape and functions of cells. Having this background knowledge, we are now able to talk about the mechanisms behind epigenetics. One of the mechanisms for epigenetics to occur is via histone modification. So histone modification gives rise to two different types of compaction, euchromatin or heterochromatin. The different compaction in histone modification may result in different gene expression. Some are expressed in one type of cell, for example the heart muscle cell, but not in the other, for example the brain cell. Expressed genes are said to be turned on, and unexpressed genes are said to be turned off. One example of histone modification that may lead to the formation of euchromatin is through acetylation of histone by an enzyme called histone acetyltransferase, HAT in short. It may lead to a decompaction forming euchromatin. Therefore, RNA polymerase is able to bind to DNA. Transcription is able to take place and the genes are turned on.
Let me illustrate to you the process of acetylation. This is the histone and this is the DNA strand, and this is the RNA polymerase. As you can see, the DNA and histones are packed together right now. The acetylation of the histone will cause the decompaction, forming euchromatin. The RNA polymerase is then able to bind to DNA to transcribe RNA and then synthesize protein. Therefore, the genes are said to be turned on.
Moving on, the example for the formation of heterochromatin is through metallation of histone by methyltransferase. This metallation could be recognized by heterochromatin protein 1, HP1 in short. The self-association of HP1 leads to high level of compaction, forming heterochromatin. Therefore, RNA polymerase is unable to bind to DNA. Transcription is unable to take place and the genes are turned off.
Let me again illustrate to you the process. This is heterochromatin protein 1, HP1. Right now, the histones are rather far apart. The metallation of the histone will be recognized by the HP1. Self-association of HP1 will cause high level of compaction of the histones and DNA, forming heterochromatin. RNA polymerase is unable to bind to DNA to form RNA. Therefore, the genes is said to be turned off. In summary, these are the key points we learned today. We learned how DNA is organized into chromosomes, and next, how epigenetics control gene expression, where genes are turned on or off. One of the mechanisms for epigenetics is acetylation of histones, resulting in decompaction, forming euchromatin. Genes are then turned on. Another mechanism is metallation of histones, resulting in increased compaction, forming heterochromatin. Genes are then turned off. That is all for today. Thank you.